Right, today we're on Chapter 15, or Lecture 15. That's going to correspond to the Bauman text, Chapter 15, or the Tutora text, Chapter 16, depending on which one you're using. For microbiology, we're going to be looking at the innate immunity. These are the physiological processes of humans that are incompatible with those of most plant and animal pathogens. So it's just things about being a human that are not compatible with these particular pathogens. Species resistance is a first line of defense because it's present at birth prior to contact with the infectious agents. So a first line of defense is going to be external physical barriers. These especially include the skin and mucosa. Second line of defenses are going to be internal. They will be made up of protective cells, chemicals, and processes that will either inactivate or kill the invaders. Your adaptive immunity is sometimes considered the third line of defense. This is going to include your lymphocytes. This is going to be when you respond against a unique species or strain, and it alters the body's defenses to act more effectively upon subsequent infection. So nonspecific responses are going to respond to a variety of pathogens, and when we talk about susceptibility, it's lack of immunity towards a particular condition or pathogen. So looking more detailed at our first line of defense here, we have the skin. In your skin, you've got your Langerhans cells, or your sometimes called dendritic cells, that are going to be phagocytic cells in the epidermis. They are also sometimes referred to as epidermal dendritic cells. The skin structure itself provides a barrier. It's going to be continually replaced, and it's going to contain tough collagen fibers that are going to help to prevent invaders from getting inside. The skin produces some antimicrobial peptides. In your sweat glands, they will produce perspiration. Your sebaceous glands produce sebum, and the sweat glands produce your dermacidins, which are going to be chemicals that have antimicrobial properties. So all of these different secretions are going to help to protect the skin. Lysozyme is going to destroy the cell walls of bacteria. It also cleaves the sugar subunits in the bacteria. And then sebum is the secretion that's going to contain fatty acids to lower the skin pH to about 5, which will actually inhibit many bacteria. Your mucosa are a less efficient barrier than the skin. You'll have stem cells in there that will continually shed and carry microbes away. The cells are tightly packed together to create a barrier. Goblet cells produce mucus. Mucus is going to provide a sticky trap for bacteria and pathogens. Some of the places will have ciliated columnar cells, like the human trachea, that's going to help to carry mucus up and prevent it from getting down into the airway. We have antimicrobial peptides that are going to be in your nasal mucus that will contain lysozyme. And then you'll have dendritic cells that are below the mucosa that will help to phagocytize invaders. Your normal microbiota, or normal flora, are going to be good bacteria that are going to compete with potential pathogens through microbial antagonism. They can do this by changing the pH. They may secrete substances that will limit growth as well. Your lacrimal apparatus, this is going to be in your eyes that produces tears. Blinking and tears help to wash the surface of the eye, so they will just mechanically help move things away. It also will contain lysozyme present in the tears that will help to target some of the bacteria. There are antimicrobial peptides in tears. You have defensins in there. There are actually hundreds of types of antimicrobial peptides in the body that will act on a variety of potential pathogens in several ways. Some of the secretions we produce: stomach acid, saliva with lysozyme. It will help to wash microbes from the teeth. Earwax, perspiration, many others will help to combat bacterial infections. Then the body also just expels microbes through secretions like your vaginal secretions, the flow of urine, peristalsis in the gut, defecation, vomiting, and diarrhea. So there are times when stopping diarrhea would actually be problematic and make the infection worse, because that is one of the ways that the body can eliminate organisms. So the second line of defense is going to come into play when the pathogen gets past the skin and the mucosa. They're good barriers, but they're not perfect, which is why we need a second line of defense. So the plasma is going to be the liquid component of the blood. So your serum has plasma with the clotting factors removed. Transferrin is going to bind oxygen for transport. This makes it unavailable for microbial use. Some microbes will respond by secreting their own iron-binding proteins. Complement is going to be a series of plasma proteins that act as chemotactic attractants and will trigger inflammation. And then your antibodies or immunoglobulins are going to be antigen-binding materials that are produced by the plasma cells. So some of our defensive blood cells, or leukocytes, is what we'll look at next. You have your formed elements. These are the cells and cell fragments in the plasma. Hematopoiesis is the formation of the formed elements. Your erythrocytes are the red blood cells. So their job is specifically to carry oxygen and carbon dioxide. Platelets are fragments of a giant cell called a megakaryocyte that are involved in the clotting of blood. The term leukocyte just refers to white blood cells. Cytes are cells, leuko is white. These are involved in protection from invaders. The granulocytes are a type of white blood cell that are going to have granules in the cytoplasm. And we have three types of granulocytes. Their names all end in phils. So basophils are going to stain blue and they'll release histamine. Eosinophils are going to stain red. They're able to do phagocytosis and they can go into the tissue. They tend to be active in times of parasitic worm infections and allergies. And then the neutrophils will stain purple. They will look like they have multiple nuclei. They can do phagocytosis and also go into the tissue. So diapedesis is when the cells can exit the blood to attack the microbes in the tissue. They do this by squeezing between the cells that line the capillaries. Our granulocytes, these are going to include lymphocytes and monocytes. So the lymphocytes are the smallest. They are going to do specific immunity. So this will include your B and T cells. Monocytes, these are the largest. These leave the blood and mature as macrophages. So your macrophages are phagocytic cells, and they're going to be involved with your second line of defense. They'll engulf bacteria, fungi, spores, dust, dead cells, a variety of things. Some of the macrophages are going to be fixed and some wander. The wandering macrophages leave the blood via diapedesis, and they can scavenge throughout the body. Fixed macrophages do not wander. They will be in specific areas. Some examples, we have alveolar macrophages in the lungs, the microglia in the CNS, Kupffer cells in the liver, your mononuclear phagocytic system, or your RE, reticuloendothelial system. These are going to be macrophages and monocytes that are attached to endothelial cells. Dendritic cells are derived from the same precursors as the monocytes, and they'll do phagocytosis and initiate adaptive immunity. So one way of getting a picture of what's going on in the body is to do a differential white blood cell count. This will look at the proportions of the leukocytes, and this can be used as an indicator of disease. Your lymphatic system, this will consist of lymph vessels and the structures or organs containing the lymphoid tissue and your red marrow. So these will contain large numbers of lymphocytes. So let's look at some mechanisms of phagocytosis. Phagocytosis is when a cell engulfs the invader. A phagocyte is going to be a cell capable of phagocytosis. This will include most of the leukocytes and their derivatives. Chemotaxis, this is the movement of a cell towards or away from a chemical stimulus. This includes microbial waste, damaged tissues, and chemotactic factors like peptides from complement or cytokines released by the leukocytes. Adherence is the binding of complementary chemicals on cell membranes. With opsonization, we have the coating of microbes with antimicrobial proteins of complement or antibodies. This is used to enhance phagocytosis, and the proteins are called opsins. With ingestion, this is when the phagocytes extend their pseudopodia around the microbe and internalize it in a phagosome. Phagolysosomes are digestive vesicles that are formed by the fusion of lysosomes with the phagosomes. During exocytosis, this is a process of eliminating undigested remnants of microbes and other materials. It's the reverse of phagocytosis. So your extracellular defenses are ways of doing non-phagocytic killing. We have the eosinophils. They'll attack parasitic worms by attaching to the worms. They secrete toxins onto the surface of the worm that can weaken or kill the worm. The worms can be quite large, so an eosinophil is just not going to engulf a worm that's several times its size. The NK cells, or natural killer cells, are going to secrete toxins onto the surface of virally infected cells and neoplasms. These are found in the blood, spleen, lymph nodes, and the red marrow, and they can attack any body cells that will display abnormal or unusual plasma membrane proteins. And then the neutrophils, these can also destroy microbes with phagocytosis using enzymes, but they can create reactive superoxide radicals and hydrogen peroxide and form nets of extracellular fibers to help trap bacteria. So they can do phagocytic killing and non-phagocytic killing. Some non-specific chemical defenses are going to include complement, among other things. Complement is a set of serum proteins that will initially act as opsonins and chemotactic factors. They'll trigger inflammation, fever, and result in the lysis of foreign cells. So 3 main steps to what complement does: cytolysis, opsonization, and inflammation. The classical pathway involves binding antibodies to foreign antigen to activate complement. The alternate pathway is when the pathogens or pathogenic products activate complement themselves. And then in the lectin pathway, mannose sugar attaches to molecules called lectins, and this begins the classical activation pathway. The MAC, or membrane attack complex, is the end product of a full cascade. It forms a hole in the pathogen's membranes. So complement is not something you want to have active all the time, so we have means of inactivating complement. Here the membrane-bound cell proteins will bind and break down activated complement proteins that will interrupt the cascade before damage can occur. Complement does play a role with disease. Inherited diseases may result in increased susceptibility to disease. This also may play a role in diseases with immune content. It's implicated to have a role in Alzheimer's and other neurodegenerative disorders. So microbes do have means of evading complement. Some will have capsules that contain sialic acid, which discourages opsonization and the membrane attack complex. Other capsules may interfere with other components. So interferon, this is a series of proteins that are released by host cells to non-specifically inhibit viral spread. They'll cause the symptoms of viral infection, so interferon makes us feel pretty lousy. Both alpha and beta are type 1 interferons with similar structure and action. So alpha interferon is produced within hours of infection. It's secreted by virally infected monocytes and macrophages and some of the lymphocytes. Beta interferon is also going to be present within hours of infection. It's secreted by virally infected monocytes, macrophages, and some of the lymphocytes as well. Gamma interferon is produced by activated T lymphocytes and natural killer cells. This appears later, and it's stimulated macrophages and phagocytic activity of the neutrophils, so it can act as a macrophage activation factor. We also have iron-binding proteins that will play a role. They keep the concentration of free iron low because it's bound to proteins like transferrin, lactoferrin, and ferritin, and hemoglobin. The importance of keeping the concentration of iron low is that it will deprive the microbes which need that iron. AVP, or antiviral proteins, alpha and beta interferon, bind receptors on neighboring cells, and they'll trigger the production of antiviral proteins that will bind the double-stranded RNA in the viruses and inhibit protein synthesis. Interferon therapy is made with recombinant DNA technology. There are some viruses that have a small response, none at all in others. So chronic granulomatous disease responds well. It's got an inability to produce gamma interferon. So the defensins, these are small peptides that will protect against a wide array of pathogens. These can damage cytoplasmic membranes or interfere with other metabolic processes. So inflammation, not fun stuff. This is a local defensive response. When it's acute, it develops quickly, it's short-lived, and typically beneficial. It usually eliminates the condition that precipitated it. Chronic inflammation, on the other hand, develops slowly and lasts a long time. This type of inflammation can actually cause damage or death to the tissues and result in disease. Signs and symptoms of inflammation include rubor, calor, tumor, dolor, loss of function, and we can look at these with the acronym SHARP. So rubor is redness, calor is heat, tumor is edema, dolor is pain, and then we have the loss of function that goes with that. Its role in defense is it's going to increase dilation and the permeability of the blood vessels. It will lead to the migration of the phagocytes and tissue repair. The dilation and increased permeability of the blood vessels is important because it allows more defensive substances in, but it also leads to edema. Bradykinin is a vasodilator that forms when clotting follows damage to blood vessels. Histamine also causes vasodilation, brings more blood to the area, which will mean more phagocytes, oxygen, and nutrients can be released by basophils, platelets, and mast cells. The prostaglandins, this is released by the macrophages using the TLRs and NODs. This will increase vascular permeability, intensify the effects of histamine and the kinins. Your prostaglandins are derived from certain fats that you eat. The omega-6 fatty acid series is used to make prostaglandins that will promote clotting and inflammation. The omega-3 series are going to make prostaglandins that are going to modulate and decrease clotting and inflammation. Your leukotrienes, these are inflammatory chemicals that are going to be released from the same cells as the prostaglandins. They'll increase vascular permeability that is produced by the mast cells. Mast cells are located in the connective tissue, and they'll release histamine when they're exposed to the complement, the C3a and C5a component. Your cytokines are secreted by activated fixed macrophages. They'll increase vasodilation and vascular permeability. Vasodilation and permeability will lead to edema, which puts pressure on nerve endings. It will also deliver fibrinogen to form a clot or wall an area off. Clots in tissue are important because they will prevent the pathogen and their toxins from spreading. So some organisms are known for forming clots, like Staphylococcus. Pus is fluid containing dead tissue cells and leukocytes in a walled-off area of destruction. Your abscess is just the isolation of a site of infection. It's that local area that's infected. So with the migration of phagocytes, this will increase blood flow, and it delivers monocytes and neutrophils to the site of infection. Margination is the process of the leukocytes sticking to the walls of the vessels, where diapedesis is the migration of the phagocytes to the blood vessels to reach the damaged area. So we have tissue repair. This is going to deliver extra oxygen and nutrients. Not all sites are repairable. So with cardiac tissue and nervous tissue, those cells do not repair well. Sites with regular cell division repair rapidly. One of the downsides is you can have fibroblasts create scar tissue. Fever can be important as well. It's a temperature above 37 degrees Celsius. It can augment inflammation. It does have some unpleasant side effects. The blood vessels in the skin constrict, causing a chill. It will enhance interferon, which will make you feel lousy, lead to phagocytosis, repair, and inhibition of microbial growth. However, if the fever gets too high, it will denature proteins, and that's problematic. The crisis is the phase of the fever when the heat is lost to vasodilation and sweating. Here, the body temperature starts to fall, and this coincides with when the infection begins to subside. Your pyrogens are chemicals that are going to trigger the hypothalamus to raise the tissue temperature. IL-1 is an example of a pyrogen released by phagocytes that have phagocytized bacteria, and it helps to increase the production of T cells.